open the...
It's fine for us.
Yes, it's...
Okay,
Wait,
brilliant.
wait.
So shall I carry on?
Wait.
Yeah.
Ah.
Yeah. You can.
Yeah. Good.
Perfect. Brilliant. Well, thanks very much for the organizers inviting me. Good afternoon, everyone. I'm Mazdak Salavati. I'm a core scientist at the Roslin Institute of Edinburgh in Scotland, Edinburgh. I am fully aware I'm standing between you and your dinner, probably. So try to make it as short as possible, not to keep you any further. But what I'm going to present to you is actually quite an old paper which we started working on in 2018, but it was based on an idea of using ATAC-Seq to profile chromatin in developing pig muscle tissues. This work was carried out as part of Dr. Emily Clark's lab, which is the functional genomics lab at Roslin. And we are involved in a variety of projects that Emily probably hinted at earlier, at a couple of them. But we basically do experiments in our large animal facility all the way to the community and presenting results and actually taking initiative on multiple consortiums. So the piece of work that I'm going to present to you will be a little bit about the publication's design, and I will touch mainly on the methodology because this paper actually turned out to be a method paper more than anything else. A little bit about the results and a take-home message. So I'll start with the end at the beginning. So this is the paper that was published earlier this year, actually, in February 2022. But originally the study, the design, and the work was actually carried out in 2018, 2019. It was written, and then it went through a peer review process of nearly 2 years, which you will see, I will touch on later in the next slides, because we had some issues with the data and we were trying to convince ourselves, including the reviewers, that this actually is what it says it is. The idea of the study was to utilize some archived muscle tissues from piglets that were collected in 2015, 2016, alongside some other samples that were freshly captured in 2018, to create a developmental timeline of muscle development in pig tissues. The reason for that is obviously is economically relevant traits in pig production, and these points were chosen as given a specific weighting. So day 45 muscle samples because the primary muscle fibers form. Day 60 of the gestation where the secondary muscle fibers start to shape. Day 90, which the fiber formation is basically ceased, and after that will be a hypertrophic stage of the muscle growth, and 1 week and 6 weeks of age, which are postnatal time frame. The paper also was mainly focused on a comparison of 2 different methodologies. Methodologies both focused on the library preparation and the sample preparation for ATAC, one of them using frozen tissues, and the other one using cryopreserved nuclei and then frozen nuclei. And the protocol, as the paper is a FANG-compliant paper, are publicly available, so feel free to follow these links or find the analysis protocols on the FANG's data portal for the comparisons. But I will show specifically a part of the result of the paper, which is focused on the comparison between frozen cryopreserved nuclei and fresh frozen tissue. As I said earlier, the experimental design of the paper was focused on a couple of timelines. So this table shows you the that were captured in each timeline and also the phenotype of the piglet itself. So we actually did capture body weight for the study, and we knew within each litter which animal was the largest, which one was the average of the litter, and which one was the smallest. So there were 3 samples for the day 45 and 4 samples for the day 60, and quite a lot of samples for the day 90 of the gestation of the tendinous muscle, and also the postnatal time points, which was 1 week of age and 6 weeks of age. And in the last 2 columns, can you see my mouse, by the way? Can someone shout, please? But does it help if I do this? Okay. So as you can see in the last 2 columns, these tissues were utilized for the ATAC-Seq and the RNA-Seq. Not all of them, but the majority of them were compatible for the 2 sequencing technologies. We had 24 samples from 5 time points and the 3 piglet sizes I mentioned for the DNA and nuclear preps, and 26 for the RNA-Seq. The ATAC-Seq analysis was carried out after sequencing these libraries on a 50 nucleotide base pair end prep using a specific software called GenRich, which is slightly different than the Match2 that Gabriel introduced. That piece of software was used to do multiple sets of peak calling. One of them was for a specific time point. For example, we had lots of samples from day 90. So we had a specific set of peaks for day 90 of the muscle tissues. And we also did the same traditional method of consensus peaks for differential expression. And that differential expression was carried out using DSeq2. We also did some hints enrichment, which is a transcription factor enrichment analysis. I will show the results on that also. We then used the RNA-seq data to do a Callisto-based transcription analysis and differential expression using DSeq2. And then we did an overlay of the DSeq2 results from the RNA-seq and the day 90 specific peaks to explore the hypothesis shaping around if there is any large versus small or average versus small or average versus large differences between open chromatin of these muscle tissues. So before I actually get into the result, there are 2 points in the paper that I wanted to highlight. First of all, the method that was used to create the consensus peaks. This is quite a tricky set of criteria that if you're doing ATAC-Seq paper, you should really consider what defines a consensus peak. In this specific study, we decided to use bed tools and merge any peak within the 10 nucleotide base to have another peak to create a consensus. And then there was a support value for it, which we decided to put a minimum 3 tissues to support said peak. And that resulted in around 12,000 peaks as a consensus. This is a very specific set of criteria that we use for this study, but my recommendation is that for any study that has done similar consensus peak calling, one should optimize the criteria required for creating this set of consensus open chromatin peaks. The second thing was another method of normalization of the counts, which was suggested in the reviewers we use, and you can see in the red box that I've highlighted the normalization counts for doing the differential expression of these open regions. So without further ado, I'll walk you through the results. You've seen this slide today a couple of times, which is great because with the introduction from Anne-Sophie and Gabriel and Jose, I can easily straight jump into why this is important. So if you're interested in the ATAC-Seq results and data set are these bits that get sequenced. So you either get segments that are the nucleus-free regions, or you potentially get the regions that actually have them inside that section of the read. And the way you can look at this is through a graph like this. So this graph shows you the distribution of the size on the x-axis and the normalized counts density or the counts of the read falling into those regions on the y-axis. And that nucleosome-free region, which is less than 150 base pairs, is quite clear in the data of the examples. And by the stretch of an imagination, you can probably see there are 2 more peaks on that distribution, which represents mononucleosome and dinucleosome probably. But again, this is something to check, as Gabriel said, is a very good quality control check for your data set before actually looking to do the ATAC-Seq data. We carefully overlaid the ATAC-Seq and RNA-seq data sets. These are just examples. GAPDH is a housekeeping gene and CASQ1 is a muscle development gene. We wanted to make sure that the reads are aligning to the gene models properly, and we're expecting to see a large opening at the end of the gene model. So for example, both of these genes are antisense, so you can see that there's a large peak of the open chromatin at the beginning of the gene and carry on across the exons of the genes as well. We're doing these overlays to make sure the data set is clean and sanity check the data. We also looked at the consensus peak problem. So on the right-hand side here in the figure, you can see the same GAPDH gene and every single sample out of those 24, and the opening of the open chromatin region that is detected and the consensus region highlighted in green as the consensus opening for the GAPDH. So we carried the QC and the quality control of the data set. And one of the things was dimension reduction to just make sure that if we can detect the pattern between the different elements of design that we have included in the study. So obviously there was a piglet size element variable and there was a time point variable. And there was also this method of cryonuclei versus frozen tissues for the seed samples. And the 2 important things that we realized in the MDS plots that actually the majority of the separation is based by time point. As you can see, the day 45 60 samples that are quite separated from the rest of the day 90 and the postnatal samples. And the second thing, which was really reassuring for us, was that preserved nuclei samples were grouping very closely to the tissue. As these two in the lab have very different levels of preparation process, we wanted to make sure that these methods can actually perform well, and it doesn't actually cause any issues if either of those methods is used for follow-up studies. Then we moved on to annotating these open regions of chromatin within the dataset that we had, and we actually thankfully managed to annotate more than 50% on average of all the samples that we had with promoter regions within a 1 kb and or 1 to 2000 kb regions, which makes perfect because these are transcriptionally active, so you expect the DNA to be open. But also we had some samples, for example, as you can see, the day 90 large pig samples, there are two there, that these samples were somehow performing differently to the others. We also looked at the distribution of these open chromatin regions given the TSS sites. On the right-hand side with the graph, you can see the curved majority hovers around the TSS site within the region, which was a good quality check for us. So once we were happy that actually the open chromatin regions and the PPARs were reasonably distributed with the genomic features, we then moved on to differential expression analysis. So if I can remind you again of the experimental design, there were 5 time points, and we used the ATAC-Seq model to look at the differential expression of differentially accessible DNA given these 5 time points. And we found out 377 regions, majority of which were actually intronic and introgenic pig muscle with significant differential accessibility of the chromatin, which was good, but unfortunately we didn't find anything within sample size to have any effect from the piglet's size of the animal itself. So the phenotype of the size was not significantly changing any chromatin accessibility. And as I mentioned, we also did hint analysis, and hint is just a transcription factor activity enrichment assay, and it's all in silico. But what is nice about hint is this graph A on the side, which shows you in the rank of transcription factors that are being active or deactivated. And because this comparison was done according to small size animals versus large size, day 90. On the left hand side, you can see 4 of these transcription factors that are severely less active in the small size piglets and obviously highly active in animals. And vice versa, there's a GNET2 transcription factor, which is a glucose metabolism transcription factor, and is highly active in the small size piglets. Although within our analysis, we realized this might be a bit of an artifact for the CBPE, which is a developmental muscle transcription factor, because the enrichment score was diabolically high. Also the FDR value. We actually reflected this in the paper, so we weren't sure if that's a real signal or just an artifact because of the type of analysis. And then looking at the RNA-seq data set, pretty much similar approach by dimension reduction to look to see if there's any specific separation of samples that matches or doesn't match the original design of the study. And thankfully, at the RNA-seq level, also the same timeline gene expression profile was quite highlighted. So you can see the day 45 samples, the 60 samples are slightly separated from the day 90 and the postnatal. So there was a clear transcriptometry there for the developmental atlas. And then I appreciate you probably would not be able to see all the genes in these graphs, but this is the differential expression analysis based on the RNA-seq, which was from the matching tissue that I've done ATAC-Seq on. And the 3 conditions that we compared them were the large phenotype versus small, average phenotype versus small, and average phenotype versus large. And just for the time, I'm only going to show you as a zoom level for the large versus small log fold change and the p-values for the differential expression. And I would like to draw your attention to a couple of genes that were quite interesting for us. So MyoG was myosin G, which is a muscle fiber protein coding gene, which was slightly operating in the large animals, large piglets within the same litter comparison to the smaller size one. Also other muscle-related genes like CASQ1 and RIR2, which are well-known muscle development genes, started showing them. So up to this point of the story, we had some understanding of differential expression within the timeline and differential accessibility of the DNA within the timeline. And also we could see that there is this large versus small difference, obviously, at the RNA-seq level. So the logical step for us was to overlay the RNA-seq data and the ATAC-Seq data to see if we can actually upload further information. And this is the type of graph that we came up with. So on the x-axis, you're looking at the genomic location centered by the TSS of the gene model. And if there are ATAC-Seq peaks on the left side of that, which means they're upstream of the gene model, and if on the right-hand side, it means they're downstream of the gene model. On the y-axis, pardon me, you're looking at the size of the peak. So for example, a peak that is very, very large means that the region that was assigned to that peak was quite a number of base pair in size. And this graph shows you the sets of, if you remember from my previous slide, that we have done a time specific and phenotype specific for day 90. So for day 90 animals, we only peak call from the large phenotypes, and these are the peaks that don't exist in small size animals. So these are almost specific at day 90 to the large sized piglets. And then we overlaid these peaks with the closest gene that we're annotated with, and we highlighted the genes that are differentially expressed in green. I appreciate this is a lot of information, but just to recap, you're looking at the open region chromatin that is annotated but differentially expressed genes, and those are the ones highlighted in green. And there weren't that many genes in the large size animals that had a specific open chromatin profile associated with them, apart from the rear two. But when we did the same analysis with a small size muscle profile and the open chromatin specific to the small size piglets, then we started seeing some interesting signals coming up. As you can see, the MyoG there, the rear two there, and also rear two there. So there are pieces of DNA and open chromatin profiles that are associated with certain genes that make tissue-specific sense that are in the small size animals. So the move forward from this point was to understand the function of these open chromatin regions, which unfortunately didn't fit in the realm of this paper. I have done some other work. Emily has basically carried on this project on further research for identifying these regions. And before I wrap up, I just wanted to show you some quality control metrics and the issues that we had. I mentioned earlier at the beginning of the paper that we had some issues with the data set. And one of the things we wanted to 100% prove was the fact that the library progression method, either being cryopreserved nuclei or frozen tissue method, it would not affect any annotation downstream of the work. As you can see here in this graph, there's no difference between any of these promoter or enhancer, or sorry, promoter and gene model regions between the two library methods, which was really good news for us. We also had the same thing across ENCODE recommended scoring systems that Gabriel briefly touched on. So for example, number of the uniquely mapped reads, enrichment scores, total reads, peaks that are called, the PCR bottlenecking coefficients, nucleus-free region, and the FRIP score that also Gabriel mentioned. And apart from the enrichment score that was actually slightly higher in the corpus of nuclei in comparison to the frozen tissue, there was no significant difference between any of the other scores between the two methodologies. This is where I was hoping to land on. So thankfully, Gabriel actually put this link up, so you already seen that there's a set of guidelines from ENCODE project and GTEx that recommend certain criteria for human and mouse-based studies that they've carried out as part of those consortium works. So there are sets of there were recommendations in terms of the depth of this. So the depth of the sequencing should be more than 50 million reads for a parent and more than 25 million for a single end. And our study was well above that, so we passed on that one. The alignment and the mapping quality should be above 95%, and so was ours. Unfortunately, our non-redundant fraction didn't match the levels of expectation that is recommended by ENCODE. They recommend 0.9, and ours were much lower than that. I'll show you in the next slide. The nucleus-free region was present in all the samples. The PCR bottlenecking coefficient was perfectly fine, and our FRIP score was also quite low. And I don't know if we were back in 2019 and did the work again, we would have used the same tools, or if there is any difference between macular gene peak color tools that creates that difference. And these are just food for thoughts. And also the TSS enrichment score was pretty much the same across the library. So we didn't manage to obtain all the quality measures that ENCODE wanted, but we passed most of them. given the circumstances of the archive tissues that we were using and the fact that this study was done 3 years ago. So it's quite an old study right now. And that's it. I would basically wrap up on a take-home message slide because I think that was pretty much the lessons that we learned going through this process. ATAC-Seq data is quite noisy and requires very careful consideration on how we do the metrics that we consider to be a guideline for standard metric. And specifically, the tool itself might have an effect. We don't know yet. I haven't actually done a comparison to see if there's a difference or not. But for example, for the reproducibility measure of the IDR, these are very different from each other. I would refer you to, for example, a guideline from Harvard University, the FAST informatics, that explains in detail why GenRich and MaxStore are such different pairs of softwares when it comes to feed calling. The library normalization method was actually useful to enable us to do differential accessibility analysis for these libraries. We are quite confident that the observation library methodology that is published as part of this paper is actually a very good standard because in our samples didn't change anything, although sample numbers are small, but it's really the result for setting this up for the thickness of the paper. And also we did find developmental specific patterns in the data, and some of them match with the differentiation of the RNA-seq as well. Unfortunately, the piglet size wasn't that specific as we wanted it to be. However, we did manage to capture some signal on the phenotype as well as the timer. And on that slide, I will wrap up as an acknowledgement and thanks to all the PIs involved. And this project was actually funded by an Institute Strategic Program, ISP fund, to Emily and Musa, Alan Ashabat, and Fabio Guanadir with PIs in the grant. And just wanted to thank all the postdocs and technicians who helped us both in the lab and in the farm to carry out the study. Thanks for listening. Happy to take your questions.
Thanks, Mazdak. I don't know if there is any questions from people here in Limoges. No, they're still moving. And on the chat box, there is some questions or not?
Not
Maybe
yet.
one. There's number one there on the chat.
Yeah, but it was before.
Yeah, I'm looking at the chat. I can't see. There's a couple of yeses. I'm assuming
Yeah,
that's because they could read me.
Ah, okay.
Just a yes.
Okay, okay.
Yes.
Yeah, people got excited, you see, when you give your talk, they say, yes, yes. You say you had quite, did you have a tough review for the paper, for the manuscript?
Yes, I think it was one of the toughest we had. So the paper was submitted on bioRxiv January 2020, and it was finally accepted in December 2021. So nearly 2 years.
Ah, well, yeah. Okay.
We persevered.
We
can go around and talk about the PTSD now in workshops like this.
Yeah. So now Gabriel knows where he doesn't need to send the paper.
No, don't think about it. You are attracting.
Yeah. Okay. Okay. So if there is no, there is a question from Isabetta. Did you record the metadata of positions of fetus in utero? The distance of the fetus to placenta may cause effect on the fetal growth.
That's a question, Elisabetta, and unfortunately, no, we don't have that as far as I know. Emily, correct me if I'm wrong. I don't think we recorded the position to placenta, but what we did capture quite thoroughly were the body weights and the.
Yeah, so we did actually. We do have that information because the samples were from a larger study that looked at measuring placental blood using a Doppler scanner. So that work was published just before our paper, I think, and did identify differences in blood that were related to size of the piglets. So, however, I don't think we've got the number of biological replicates in our study to actually statistically link the two studies together. But yeah, so there's that information.
Sorry, I was wrong. Thanks for correcting me. We do have that information. We use it.
Yeah, we do.
And Elisabetta replied, and she said, thanks to Mazdak and Emily. So, okay, good. So I don't know, I don't think will be any other questions, or maybe people online can express themselves directly, but I don't know if there is any who wants to say or add something. No? So if not...
There is one.
Ah.
Do you combine some GWAS results to find some candidate genes?
Yeah, I don't know if Mazdak, you can see in chat box.
Yes, I can see. So I think I briefly touched on this. So yes, this study went on to be a bit of a pilot for another body of work that Emily leads with some other colleagues here at Wesleyan. And definitely they are looking into those phenotype-specific regions, the accessible that is phenotype-specific, and the underlying variants that could actually not only inform doing GWAS, but also look retrospectively on other GWAS sets to see if there's any specific variants associated with that. That's an ongoing work, so I don't have any results to share with you, unfortunately.
And I suppose Gabriel can, or maybe Gabriel can add something, and I suppose you are doing same for the Bovrek project.
We are planning.
It's enough. Okay, so if no more questions, maybe I count up to 3, and after we'll be finished. 1, 2, 3. Okay, nobody else. Okay. No, it's been quite a long day, especially for people here. And first of all, I want to apologize again for the technical issues we had. We had some. And I would like to thank the local organizing committee, Véronique, Nathalie, and Lionel, who are set up for today, but also for the coming days, this program and all the logistics behind it. I want to thank also your IT colleague who show up many times when we have technical issues. And I hope you enjoy the talks of this afternoon, and you learn things, especially about our nice Bovrek project. And so we will carry on the workshop. Tomorrow will be only for people attending in Limoges. It will be a lab work, and we will try to record it. And on Wednesday morning and Thursday morning will be the bioinformatics session on the analysis of ATAC-Seq data. Probably it won't be possible to record it, but as Jose said, there will be some tutorials or documents that will be available, and we will share through our Bovrek website. Okay? So thanks a lot all, and I don't know where you are all based, people attending online, so I don't know if it's good evening, good afternoon, or good morning. So I hope you enjoyed this half day, and maybe we'll meet again in another meeting or somewhere else. Okay? Take care. Thanks a lot. Bye-bye. Bye-bye. Okay.
Thanks, guys.
Thanks. Up.